Dr. Jason Kovacic, who is the Robert Graham Chair and Professor of Medicine at the University of New South Wales, and also the Executive Director of the Victor Chang Cardiac Research Institute in Sydney. He also holds a professorship in medicine at the ICahn School of Government at Mount Sinai, or School of Medicine at Mount Sinai. Um, and I also learned that he was an internationally competitive rower and went to college on a rowing scholarship. And of course, if you've met Dr. Kovacic, this doesn't come as a surprise. And today he's going to talk about identifying a network of genes that drives FMD. So to
Right.
you, Jason.
Thanks so much. And it's a real pleasure to be with friends and have the opportunity to do this and to show you some of our work. Um, so I'm going to present a story that really goes back about 15 years. And Sandy gave a wonderful overview and a coverage of all the genetics and things we've learned. I'm just going to present one story that comes from a paper we published a few months ago. And it goes back about 15 years to when Dr. Jeff Olin and I were setting up a study at Mount Sinai in New York, and we're thinking about, back at that time, 15 years ago, what do we know about FMD and how could we go forward and design a research study? And back in about 2010, there really was very, very little known about FMD and the possible causality of FMD. We had no information on genetics. And really the data went back a few decades, and we knew that FMD did involve these changes in cells, changes in collagen deposition, and potentially changes in fibroblast cells. And those changes in fibroblasts and also smooth muscle cells that Dr. Ganesh was talking about dated back to some studies that were from the 1970s that actually showed that there were changes in these myofibroblast cells and smooth muscle cells here. And you can see these high magnification electron microscopy views of these cells showing that there were these major derangements in cell phenotype here. So, and it's no surprise, it's called fibromuscular dysplasia. It probably does involve these fibroblast cells and also changes related to fibrosis and some scarring. So we set up this study called the DEFINE study. We started work on it in about 2011, and Professor Olin and I initially set out to enroll about 200 people. And we got the first funding to do this from philanthropy and brought the first patients into the subjects in very late 2012 and then 2013. And to get fibroblasts, we use this technique that Dr. Ganesh also uses in her studies from taking a skin biopsy. And this is from the inner upper arm. And I know there's many of you online listening that are in this study and have got had this skin biopsy taken from there. It's about a 2-3 millimeter punch biopsy, the same as a dermatologist does. We get these tiny pieces of skin. They can be grown in an incubator in the laboratory, and then we can study the cells in various different ways. So it's very, very important for me to acknowledge Dr. Valentina Descamarade, who I guess is listening in now, who's done pretty much all of the cell handling and the cell culture work. And it has been a team effort. Annette King and Daniela Katie and Dodoff were helping to collect cells, and Valentina has dedicated pretty much the last decade of her career to this study. So what we can do is, this is a piece of skin. This is looking at a high power microscope view of these cells growing in the dish. This is a piece of skin here in the lower right corner. And all of these are individual fibroblast cells that grow out from this skin over about 4 to 6 weeks. And we can then do various different things with these cells. We can harvest them. We can look at how they move, how they proliferate, their size and their characteristics. But one of the main things we were interested in doing is harvesting these cells and collecting the RNA from those cells, and then doing RNA sequencing. Similar techniques to what Dr. Ganesh was talking about before with DNA, we can also do similar things with RNA and then look at the gene profiles of these cells and quantitate upwards of around 50,000 genes in these cells. So the study design had patients and controls. So we enrolled only people with multifocal FMD because we didn't want to make things too complicated with potentially focal and multifocal, and enrolled matched controls, carefully matched controls that were matched by gender, race, ethnicity, age, smoking status, BMI, and blood pressure medications. And we grew these cells, and this is it looks like a sort of refrigerator, but it's a refrigerator that's not a refrigerator. It keeps the cells at 37 degrees at body temperature. And these are all plates of cells from people in the study. And we originally set out to do 200. We've now way exceeded that because of the success of the study. We continue to recruit and enroll into this study today. We're now up to about 450 people in the study, which is fairly even between cases with FMD and controls. And Valentina's just done an amazing job with the study and collecting not only fibroblasts, but also DNA, serum, plasma, and other biological samples. And one thing that's a little bit different to what Dr. Ganesh presented is that the kind of design of the study and the way we actually went about the processing these samples and conducting the analysis was to try and understand links between genes and how they crosstalk and interact with each other to create what we call networks of genes that can be involved in biologic processes. And it's a complementary approach to many of the things that Dr. Ganesh was talking about. So we were trying to identify these networks of genes, and they really are, it can be thought about like a sort of stacked up pyramid of genes, where there are some apex genes right at the top that control a whole bunch of genes down below that have all the biological effects. And these networks of genes that have been known about for decades, but only really recently been able to be understood, these can range in size from 20 or 30 genes up to thousands of genes, even tens of thousands of genes. So this is the full complex design of the study, and I won't explain all of these terms, but just to highlight that we took only females in this study, about 83 cases and 71 controls. We did a skin biopsy, got DNA, and then fibroblasts for RNA and blood for DNA. And then we ran this through quite a complicated pipeline of analysis. And I won't explain all of this to you, but just to say, one of the core objectives was to find these networks of genes. And we did find this one that we called Super Network A. And then we basically spent several years understanding how that network causes disease through various different experiments, including work in mice. So we actually found about 23 different networks, but one network in these fibroblast cells was overwhelmingly linked to FMD. And we linked it to FMD through multiple different statistical analyses and approaches that included leveraging some of the data sets that Dr. Ganesh spoke about and additional work. And this is a sort of summary of the key genes in the network. It's not all the genes in the network, but what you can actually see is these 14 genes here, there's 10 of them here and the 4 around the periphery, that were what are the apex genes of the pyramid that I was referring to before. These are called the driver genes that control the whole network. And one of these key driver genes called UBR4 was a very, very appealing candidate to use to study this network. And I just highlight for a brief moment that what we linked to the disease was this network, and then we took UBR4 as our tool to modulate the network. We did a lot of work to look at the UBR4 gene and protein, and these are just human samples where we looked at the internal mammary artery, which is an artery that runs behind the breastbone, but which has been well described as can develop FMD. And this is the human renal artery, also obviously a key site for FMD. You can see this UBR4 protein has very robust levels in the wall of these arteries, and it coexists with, in this case, smooth muscle cells. So we went ahead to create mice where we took out this UBR4 gene, knocked it out in these smooth muscle cells in the wall of the artery. And this is a staining picture for UBR4, where UBR4 is in red. And here we've knocked it out, not entirely. In fact, it's still present in these endothelial cells, but you can see there's dramatically reduced levels of UBR4 in the wall of the artery. And so what did we find with these mice? This is now summarizing about 4 years of work in a couple of slides, but we grew and aged various cohorts of mice that went out to over a year of age. And after a lot of work, we found that the mice had significant enlargement of the major vessels. There was enlargement of the abdominal aorta, and you can see there the significant enlargement. It's about 50% wider. You can see if we look at the aorta, part of the aorta that's in the chest of the mice, this is the heart down here. This is the aorta coming up, and this is one of the carotid arteries going up to the brain. And this is in a normal mouse. And you can see here significantly larger aorta in the chest of these mice across all these different places that we measured. And here we're doing looking at the size of the aorta and different features under the microscope. And you can see here, this is in the mouse, it's a circular size. But it gets squished down as we actually prepare these slides into this sort of flat donut thing. But you can appreciate that the length around here, the internal perimeter is significantly longer. The outer perimeter around the outside is longer. The actual thickness of the wall is no different. But if you stretch this out and made it back into its normal sort of circular shape, there's a significant increase in the size. And one thing that Dr. Ganesh said in her slide of that human carotid artery that had resected in the theatre, where there was a loss of the normal squiggliness of the wall of the artery, we see exactly the same thing in these mice arteries as well. So did we see string of beads? No, we didn't see string of beads, but there are just incredibly important differences between mice and humans. And we never actually expected to see the FMD string of beads because the blood flow rates, the heart rates, the shear forces, and so many other things are so different between mice and humans. But we do know that larger aortas and aneurysms are key features of FMD, and that's pretty much what we found in this mouse model. So what was also really important to us is trying to then understand what this network of genes is actually doing and how the network is causing FMD. And Dr. Ganesh was already saying that FMD does involve changes in collagen and matrix. And collagen and matrix are critical in actually controlling the architecture and the integrity of different tissues in our bodies. Collagen and matrix are responsible for the elasticity of our skin and also the elasticity and the integrity of the artery wall. And so we did suspect that might be the case. And this is just one experiment I'll show you where we knocked out UBR4, reduced its levels here in cells in the dish, and looked specifically at these collagen and matrix proteins. And this is what's called a heat map, where red is higher levels of these proteins and blue is lower levels. And you can see that multiple of these collagen and matrix proteins, this is collagen 5A2, collagen 4A2 that Dr. Ganesh was talking about, collagen 3A1, fibrillin 1, many of these proteins that we know are linked to FMD and SCAD also are deranged when we upset this network by changing the levels of UBR4. So I think this is actually very consistent with our understanding of these diseases now, is that multiple of these collagen and matrix proteins can be perturbed that lead to FMD and SCAD. And when we found this network that's linked to FMD, it reassuringly perturbs many of these collagen and matrix proteins that are linked to this disease. So just to summarize the findings, we identified this network that's strongly associated with FMD. We found and validated that UBR4 controls the whole network. When we knocked out or removed the gene of UBR4 from the cells in the wall of arteries in mice developed some of the features of FMD. And we found that these enlarged arteries in mice were mostly because of changes in collagen and matrix. And I would also say another key thing that we did with this study was we've actually now developed the first mouse model for FMD, which will allow a whole lot of new possibilities for studying and understanding FMD using different approaches and tools. So all of this got published late last year in one of the Nature family journals. And I'd again shout out to the whole team here, particularly Valentina that led the study, but everyone, there's several on this call. Professor Jeffrey Olin, a great colleague and friend, was instrumental in setting this all up, but it took a team of people to achieve this. And just to acknowledge those people now, Jeff, Daniela, and Annette, who led the clinical side, Valentina, Angela, Bargravi, and Yang from my lab at Mount Sinai, that are still working on this disease and this UBR4 gene, all the bioinformaticians, my lab in Australia that didn't have anything to do with this study, but are still working on actually PRACTA1 at the moment, and all the other collaborators I have the pleasure and honor of working with, including Santi Ganesh, who's on the call here. So thank you very much, and I'd be happy to take questions with Dr. Ganesh.